Good morning. My name is Eric Klaassen. Very welcome to this and yet another session, scientific session of Yakult on the gut. In this case, we will talk about the gut-brain axis. Now, why the gut-brain axis? Well, again, we are doing this partly online because of COVID, and as you might know, COVID also has induced a lot of stress. So the whole stress discussion has become very relevant, both in students and people who work at home and in other settings. So I think you would really relate to the fact that stress and the brain have become more important to these recent developments. But scientifically, the gut-brain axis has been a field of study for the last 10 years at least that has gotten an enormous amount of attention. And Yakult is on a mission to educate the general public on how they could change their behavior and attitudes and diet to give more meaning to the gut-brain axis. This education is, of course, not done directly, but through the media and journalists and blogs. And that's why you are so welcome today to get first-hand knowledge of three experts from the field on the gut-brain axis and on recent developments on the gut-brain axis. So, number of household issues. The questions you can pose through the Ask a Question button that you see online. Unfortunately, not all questions can be answered in view of time, but all questions will be answered on the email afterwards, as for new questions that you can also ask to the organizers. You will get a copy of all the slides in a PDF format. You cannot just use those slides. You have to ask copyright permission to the owners. So please be aware that they are for personal use only. And if you want to use anything from the PDFs, you have to ask permission to the owners. But that would also mean that with the PDFs, you don't have to take a ridiculous amount of notes because you will get all the slides. At 11:45, we will have a short bio break because the gut-brain axis also has to have the flow, so you can take care of that at 11:45. And after each presentation, we will have short question sessions, and then at the end we'll have a wrap-up with all the speakers and try to answer the remaining questions. Today, my name is Eric Klaassen. I'm an immunologist by training. I've been working on the mucosal immunology since 1980 and the role of lactic acid bacteria since 1987. But my true job is starting companies from universities on infectious disease diagnostics. So we've been very busy with COVID, but also with all other zoonotic diseases in general. Contrary to what you might think, the boy is infecting the pig here with influenza, so it's actually the pig that has a problem. From the top, the brain, to the bottom, the gut-brain axis is very complex and only has been a topic of study in the last decades. And especially the role of the innervation of the gut, so that means all the nerves that go into the gut, means that the gut is very well established as our quote-unquote second brain because of the enormous amount of nervous tissue it contains. And the diet plays an important role in modulating how the gut behaves, as we will see today. Recent studies also show that metabolic syndrome, inflammatory diseases, allergies, neurodevelopmental disorders, etc., play a very important role in the gut-brain axis. So the signaling goes both from the brain to the gut and from the gut to the brain. And you might think, and people have claimed this, I think it's not completely true, but that the gut microbiota, so the organisms that live in your gut, are at the intersections of everything. If you believe all the papers that are out there. Interestingly, both the WHO and the FAO have linked a disbalance, we call that the dysbiosis in the gut, to several diseases. Now, this is not a causal relationship, but more a correlation. So enormous amount of work still needs to be done, but in this recent Cell paper, which you will get in the PDF, you can't read this at all, I thought it was really flabbergasting to see how many correlations with how many disorders there are at the moment under study, to see whether we can link the clinical effects with this dysbiosis in the gut microbiota. So we are really making progress there. Now, today we could easily say that the social isolation of COVID is one of the big issues at the moment in society. But in this table, there are other factors that in the modern world that also can have a huge influence on sterile inflammations in your body. And these are again linked with inflammatory markers, of course, and can be linked and correlated with depression. How does that work? Well, today 3 experts will tell you how that interaction of the gut content, here on the top of the slide, this is where your food is and where eventually you make your poop, and the bottom, the inside of your body. Today's experts will explain how that relates with the gut-brain axis. Generally, we would say that there are a number of issues that you should be aware of. One is, of course, if you have a filled gut, like here the metro in Kyoto, if you have a filled gut, that it's difficult for hooligans to get on. And after, for example, antibiotic treatment, it would be very easy for hooligans to get on. So it's important to keep your gut filled. That's one. So you have a competition for space and a competition for food and a competition for adherence. But the normal bacteria in your gut will also produce, like most bioterrorists do, a plethora of compounds that on a short distance are antimicrobial. So your typical lactic acid bacteria in your own gut will make not just lactic acid, but also hydrogen peroxide and alcohol, etc., thereby having a positive effect to keep out the hooligans in your gut. So we view our host bacteria as our allied forces because of this colonization resistance. So it means that it's difficult to step on if there is no space and no food available for these toxic compounds, but also because of their role in the immune response. And that is, of course, something that will become clear in the coming years, because there are also an enormous amount of studies now involved in COVID and gut health. Gut health is typically linked also to your diet, and we published a paper in Frontiers in Pharmacology in 2014 showing that some Mediterranean-type vegetables like garlic, leek onion, artichokes, asparagus have a very beneficial effect on bacteria, and thereby that explains that it's not just red wine and olive oil, which is delicious and good for you, but also the typical vegetables you find in a Mediterranean diet. That's not new. Fermented foods as a dietary source of live organisms is very well described. Typically, there are cheeses like the Emmentaler cheese that contain up to 1 billion bacteria per gram. So, but the range widely varies, as you can see here, from 100 bacteria per gram to approximately 10 billion. That's a huge range. So food is not just any food, but especially also specific fermented foods, and even then there could be a wide range. So you have to take care. If you look at, for example, biological Emmental cheeses, you're not always sure that they have these huge amounts of Propionibacteria, which are the probiotic there. In conclusion, if your gut-brain is in balance on the right side of my slide, You seem to be healthy. I don't think there's anybody 100% healthy, but that doesn't matter. And if they are in a disbalance, then this will be associated with disease. And the human microbiota, therefore, both in health and disease, has a huge impact on how we function. And of course, what we can actually do about that in daily life. So that as a very general introduction, I will not take questions on this except for at the end of the meeting of today. Because, again, I'm here only for the presentation today, and I'm very, very happy to present the first speaker, Professor Dr. Jeroen Raas. And Jeroen will tell us something about the science behind the diversity in the gut microbiota, the factors that influence the diversity, and what happens if this is disturbed, and also whether changing that diversity can possibly act as a treatment for certain diseases. So it gives me great pleasure, Jeroen, to give the floor to you. Jeroen, the floor is yours.
Thank you very much, and thank you very much for the invitation and the excellent introduction that make my life a lot easier. Good morning, everybody. My name is Jeroen Raas. I'm from the KU Leuven, University of Leuven in Belgium, and also the Flemish Institute of Biotechnology. And I'm here to talk about some of the work that we do using large-scale population cohorts to study the relation between the gut and various diseases, and especially in this particular case, the gut-brain axis. Next slide, please. So this was also part of the introduction. This is why we're all here. The last 10 years have been a roller coaster in the field, where we find associations, correlations between the microbiota, changes in the microbiota, disruption of the microbiota, and a wide range of diseases. And also within the area of the gut-brain axis, so all areas of neurological disease or behavioral pathologies, we find more and more associations. Now, next slide. What we do in our lab is we use population cohorts to study these things. And we started a cohort many years ago now, which is called the Flemish Gut Flora Project. It's basically a cohort in Flanders, the north of Belgium, of more than 3,000 individuals, which we follow and study the health of these individuals, and at the same time follow their microbiota composition with a specific focus on the gut microbiota. Now, in the first study with this cohort, we first wanted to identify what are the factors explaining the variation. Why is my microbiota different than yours? And in a nutshell, we found a whole range of variables, but some of the more important are transit time, so the time of flow of food through the body, age, gender, BMI, diet, and use of drugs such as, for example, antibiotics. Now, we also use that cohort to study the gut-brain axis. Next slide. And in that cohort, we gave people a questionnaire, which is the RAND questionnaire. And with that questionnaire, we could actually assess the well-being of the participants in our study. Please note, these are all healthy, normal individuals. And so this is a snapshot, excuse me, through the population of Belgium. And so with this RAND questionnaire, we have an idea about how people's mental health, mental well-being is, and at the same time, how their physical health is. It is a subjective questionnaire which participants answer themselves. Next slide. And so we found, interestingly, quite a number of associations between the parameters of this RAND questionnaire, so a scale of how happy people felt or how they assessed themselves, their physical health, and the microbiota. We found strong associations between certain bacteria, you see the names here, Faecalibacterium, Coprococcus, Dialister, and mental health. And so two bacteria were associated with higher quality of life, and furthermore, two organisms were associated with depression in a negative sense. So we have a diagnosis of depression in our cohort, and people who are depressed, they have less of these bacteria. So that's very interesting, because then conceptually you can think, well, if people who are depressed have these have too little of these bacteria, why not give this back, right? That would be an interesting concept. More about that later. Next slide. Another thing we do is technological innovation, and we've found an approach which we call quantitative microbiota profiling. It's a whole mouthful, but in essence it means that instead of measuring the microbiota in relative abundances, saying, like, for example, of your gut microbiota, 20% are these types of bacteria and 30% are these bacteria, we can actually determine the number of bacteria per gram of fecal material, because we work on fecal material to study these communities. And this gives very important advantages, because it allows us to very precisely measure quantitative differences between individuals. And one of the surprises of these studies was, for example, that there's actually a very big difference between individuals. There's people who have a lot of bacteria in their guts, and there's people who have up to 10, up to 100 times less bacteria than other people in their guts. So the question very quickly became like, okay, so why is this relevant? Is that importance different? Can we do something with that clinically? Next slide. And so we apply this in a wide range of diseases. And this, for example, an example of Crohn's disease. Crohn's disease is a disease where for a very long time it was already known that the gut microbiota is implicated. And there were proposals of biomarkers for diagnostic testing in Crohn's disease. Now, what we found was that these biomarkers, who were developed in relative terms, if you would look at that in absolute terms, many of these associations were not true. So people would have, relatively speaking, less. For example, here in this case, people with Crohn's have more Bacteroides in relative terms than others. But if you would measure that in absolute terms, they would have exactly the same amount of Bacteroides. So therefore, it's very important to do this accurate measurement as we do. Next slide. Another thing that we apply this on is to the concept of enterotypes. Now, enterotypes is a technique we use to stratify people, to basically catalog individuals in different types of microbiota. And so we discovered this already 10 years ago, where we identified 3 types of individuals with different composition of the microbiota. Now, these 3 types are generally considered to be all healthy. It is just the difference between different people. Next slide. Now, interestingly, thinking in this in a quantitative way and using the quantitative data, we discovered that there was a 4th type, and this 4th type was really different from the 3 normal types that we've identified, because these people had a lot less bacteria, like 100-fold less. So these are people that have a very poor microbiota. We actually, at this stage, consider that microbiota as truly dysbiotic. And we call that the B2 or Bacteroides 2 enterotype, because it's similar to one of the 3 enterotypes, which is called Bacteroides, but it is disturbed and has a much lower density of bacteria. And for example, we found this to be associated with Crohn's disease. If you look at people with Crohn's disease, about 80% of these individuals have that particular type of microbiota. Next slide. And then we started looking in other diseases, and to our big surprise, we found that that particular type of microbiota is actually associated to a wide range of diseases. We found it associated to ulcerative colitis, which is another intestinal disease, or primary sclerosing cholangitis, all big names, which is an inflammation of your bile duct. And we found an association with certain subtypes of multiple sclerosis. So we see that this type is present or increased in a wide range of diseases, and that makes it very interesting as a potential more universal diagnostic. Next slide. But the biggest surprise, and very relevant to this seminar, is that we also found it associated to well-being and quality of life. So for example, on the left, you see that if we have a quality of life score, and we compare it between the 4 enterotypes, the Bacteroides 1, 2, the Prevotella and Rhuminococcus enterotypes, we see that the people with the B2 enterotype, they actually have a much lower quality of life score. Also, when we compare control healthy individuals with depression individuals, we see in red here a marked increase of the B2 enterotype. So in the population of depressed individuals, we find a higher prevalence, a higher percentage of B2. So there is an association between this enterotype and the gut-brain axis. Next slide. Now the question is why? And one of the things that we discovered, and don't have time to go into a lot of detail, but we find is that Bacteroides 2 is associated to inflammation. So if you have intestinal inflammation, even if it's low grade, so not to the extent of a Crohn's disease patient or something like that, already there we see a higher prevalence of the Bacteroides enterotype. And now we can go back to these to the field of gut-brain axis and the area of depression. We know that depression is often associated with neuroinflammation, inflammation of your nerval endings. Now the question is, perhaps there's a link there. If you have maybe some inflammation in your gut, which we notice by the B2 enterotype, maybe it is also linked to a more systemic inflammation and inflammation of your whole body, and with that also your neural system. And then the question is what's the relation there? Is the microbiota just responding, or is it actually part of the cause? And then I would like to refer back to the results that I showed you earlier. Next slide. Where, if we look at what bacteria are decreased in people with depression, these are bacteria that are so-called butyrate producers. Now, butyrate is a chemical compound that is very important in our gut. It is food for our gut cells, for the host cells, and at the same time, it's a compound that decreases inflammation. It dampens inflammation. So we're seeing in depressed patients that they have less bacteria that dampen the inflammation, suggesting that it might lead to a cause of more inflammation in the gut, which could be then linked to neuroinflammation, which could then be an explanation for the depression. But what I just said here, I have no proof for that. It is a theory, right? Because we only have a cross-sectional data set, so we only have an observation. We don't have any causal evidence at this moment. Next slide. And it's not the only interpretation, right? The inflammation is a possible route, but we know that the gut also is a production site for a whole range of chemical compounds. That all these bacteria, they make all these types of chemicals, and many of these chemicals might actually also interfere with our nervous system. And therefore, we basically also developed a technique to detect that, and we found, next slide, that very many, or the large majority of gut bacteria are actually able to produce or metabolize neuroactive compounds. So these are chemicals that are able to interact with our nervous system and our brain. And the fact that many of the bacteria in our guts can do that is also part of the reason why we think that there's such a thing as a gut-brain axis. If some of these compounds can reach the brain, and there's many routes to that as well, you'll see more of that later, then there is a potential causal interaction also possible between the bacteria and our nervous system. Next slide. So the question is, what can you do with that knowledge? Can we modulate the microbiota to treat diseases? Next slide. So here's another interesting finding. This is work that was recently published, where we found that B2 enterotype that I just said, it also increases with BMI. So people who gain weight, in the higher weight categories, we find more people that have that B2 enterotype. And again, we find it linked to people having higher inflammatory markers, for example. And so on the trajectory towards, for example, metabolic syndrome, we actually see that increase of B2. But the interesting observation came when we started looking at the drugs of these individuals. Next slide. here on the left, the top left, you see indeed that people that have a low BMI, low body mass index, they have a small percentage of Bacteroides 2 enterotype. There's only 5 to 10% of the population with a low BMI that have a B2. That is normal. That is what we expect. And if you have this increase in obesity, we see that the percentage of B2 enterotype increases. So that is expected. Now, what was the surprise? And if you could click one more time. Next slide. Yes. So what you previous slide. What you see is could you go? Yes. What you can see is if we looked at what these people took as medication, they often take statins. Statins are cholesterol regulatory drugs, and so people that were on statins, even if they had a high BMI, their percentage of Bacteroides 2 was low. So we started to speculate, and again, this is speculation, that statins could actually be a modulator, that statins somehow could be protective against the development of this B2 enterotype, this dysbiotic enterotype. And so this is a way, again, this is something we still need to prove. We don't have the strong evidence for that yet, but it shows that with these cross-sectional studies, these population studies, you can actually discover novel drugs, or in this case, existing drugs that can help on modifying the microbiota. Next slide. Now, another example of microbiota modifying strategies is that through probiotics. And so you know the story of the lactobacilli and the classic probiotics, but there's a whole new movement happening where a new type of probiotics is being developed, namely by sourcing bacteria from our own bodies. And this comes from the area of fecal transplantation, for example, where we see that transplanting fecal material from one individual to another can be used in certain diseases. Now, and if you follow that idea, it means that if you can isolate the good bugs, you could actually use them as potential future medicine. Next slide. And so that is something that is still very active in the field. And one of the things that we're realizing is that we're doing a lot of microbiota analysis and sequencing and high throughput omics and systems biology and whatnot. But the answer, but one of the things that we're realizing is that the organisms themselves that live in the gut, we need to study them in the lab much better, and we need to spend a lot more time isolating those organisms from the gut. Next slide. And that is something that we do in the lab, and we have now a very large culture collection of over 3,000 gut bacteria that we have in the little vials that you see there. And these bacteria we can now use to study and hopefully also use to develop novel probiotics. Next slide. And this is the final slide of, and this is the hope, I would say, for the depression study that we're doing. So I told you that we were missing bugs in depressed individuals, and I told you it would be really nice if we could give them back. But in order to do that, of course, we need to do experiments. And so for that, we are now collaborating with a team in Cork of Jared Clark and John Cryan to do experiments to see in models whether bringing back these organisms is actually beneficial for people. Now, next slide. I think I'd like to give you a few take-home messages. I think I would like to convince you that these population cohorts are actually very useful to identify important novel clues on how the microbiota is related, but we have to be careful with causality. I've shown you that quantitative analysis can give you very interesting insights, and for depression, I think we're finding very interesting clues, but we still have to be a little bit careful with causality. We still need to do the experiments in which proving that changing the microbiota can actually be a treatment for depression, because that's something we have not shown yet. Next slide. And this is the people that do all the work. I'm sitting here giving talks, but this is the people in the lab and on all our collaborators. Next slide. And we do a lot of interactions also with industry and funding from European and national funding. Next slide. And with that, I'd like to thank you for your attention.
Great. Thank you very much, Professor Raas. I immediately have a question from the audience, and that I like the question a lot because it says, does the B2 enterotype change if a person is not depressed anymore?
That's a very good question,
Yes.
and I don't have the answer to that yet because we don't, at this moment, we don't have longitudinal studies, or we have not performed longitudinal studies yet in which we track people in time. That it is very interesting, and we're setting up and doing many of these studies as we speak, but we don't have such data yet. I would, if there's a link between inflammation and depression, if that is really true, and that link is bidirectional, because that's also not certain, then you would expect indeed that the B2 enterotype would reduce. Or you would say that the person does something, for example, change their diet, the B2 gets shifted away, and if the B2 would be causal to the depression, if it would be causal to the depression, then this would lead to perhaps a reduction of depression. Now, at the same time, I want to be a little bit careful in what I'm saying. There is a gut-brain axis, and I'm convinced that we will be able to develop things in that area, but depression is a multifactorial disease, and there are other things that also play a role in that disease, so it's not only the gut. The gut is part of the story, but there's more to this as well.
Yeah, that also means there is no one-size-fits-all solution. But talking about that, and you mentioned it yourself in your explanation, there's another question linking to that, and that is how long do you need to adjust your diet in order to see changes, let's say relevant changes in the microbiota?
Yeah, that's a very good question. So we're doing a lot of experiments as we speak, actually, with dietary interventions to see how we can modulate the microbiota and what type of dietary interventions we should do. Because the honest truth is that area of the field is still very much underdeveloped. We know that changing your diet can affect your microbiota. We know it can actually happen very rapidly, within days. But the targeted changes or changing of the microbiota, pushing the microbiota deliberately in a certain or another direction, that is a lot more difficult. And people, it has been shown that people respond very differently to dietary changes. So it's probably personalized. But just to answer on the time question, I think it can go actually very rapidly. In a matter of days, you can really make a shift.
Great. Then another question, what does serotonin in your gut do to your mood?
Yeah. That's a very good question. So there's a lot of production of serotonin in the gut. Yeah. I'm going to be very careful because I can't really say that it has a very strong causal effect on mood, just like that. There's a lot of pathways involved there. There's also cross metabolization of these organisms. But indeed, there is production of serotonin, and I think it should be studied a lot more. And I know this is not a very satisfying answer, but I at this moment I don't want to speculate too much about it.
We'll come back to that later in the wrap-up, because I'm sure Robert Jan Brummer has something to say on that. By B2 being present in several diseases, do you mean several autoimmune diseases or on a more broad scale, next to depression?
Well, that's a very good question. So the ulcerative colitis, Crohn's, MS are typical autoimmune diseases. But on the other hand, metabolic syndrome is not, obesity is not, depression, I don't think it is either. So it is not autoimmune disease, but I do think it is all related, or a lot of it is related to inflammation and or disturbance of the gut environment. And so I think that's the common denominator between all of these diseases.
Yeah. I have another question, but I'm going to answer that myself, Jeroen, if you let me. The question is, I read somewhere that you should not take probiotic supplements if you are on antibiotics because your body has to take care of recovery itself. Is that right? I would like to answer that because this is a field that has been addressed by, especially by Ivan van der Plas, I think, in earlier studies like almost 15, 20 years ago, where Ivan showed that most probiotic supplements, as you can buy them in Belgium, because Ivan is Belgium, they contain probiotic strains that are very well capable of pumping antibiotics out. And so that means that on the commercial probiotics, you could take them hand in hand with the antibiotics. That would not damage the probiotic, but actually give the desired effects immediately. This is not true, of course, for Saccharomyces boulardii. That's a yeast that does not have such an antibiotic pump, but it's true for all the lactic acid, most of the lactic acid commercial products. Yes, if your diet consists of prebiotic fibers and you have enough prebiotic fibers and you don't get too much sugar, then your body can easily recover from an antibiotic insult. Now, usually you get your antibiotics for a reason, and that reason might also impair your normal diet and especially the variety in your diet. So in most settings where you use an antibiotic, it might be useful to supplement with a probiotic, but if your diet is completely up to snuff and you don't use too much sugar, which actually favors the quote-unquote pathogenic bacteria, then the probiotics should not be necessary. We can all comment on this later. The next question, Jeroen, is do you see associations between illness, the microbiome, and the diet if you look at, for example, vegetarian, vegan, or carnivorous diets?
I don't think the triangle has been made as a study. A lot of the arms of the triangle have been made. We see effect of meat and fruit and vegetable and fiber consumption on the composition of the microbiota. There is shifts in the microbiota associated to disease and illness, and there is shifts on the other side of the triangle. There's associations between, for example, with meat consumption and illnesses such as cardiovascular disease, and so or cancer. But a study that has made the link the causal link really between those three triangles at the same time, I don't think that has been properly done yet, or at least not to a large extent.
Yeah.
So it's very interesting to do.
Yeah, good point. Then we have a question from Munich. Herzlich willkommen. And the question is a statement: giving back good gut bacterial strains is a nice idea, but it isn't a big problem because they won't stay in the gut and be eliminated immediately. That's an interesting point. Maybe it's not for a scientific discussion, it's more for a commercial discussion, because the fact that indeed bacteria that you do not have since your early life are eliminated relatively fast and don't colonize. That's the way we would describe that. Although in Finland you now see that pregnant women have been using LGG for a long time, and these babies constitutively express LGG. So, but the commercial aspect here is, of course, that indeed, if you take your probiotic every day, that's commercially very attractive, but of course, for the user, not. Lots of studies, maybe Jeroen can also elucidate to that, I'm sure Robert Jan will. Lots of studies are now being done, also in the Netherlands, on babies, where you will give babies probiotics, and of course, if you give them early in life, in the first months, then they will colonize, and they will be later on, with the right diet, be careful there, with the right diet, they will later on in life be constitutively expressed, and you will take them with you for life. So it's a good remark, but I don't know whether it helps us now. Jeroen.
Sorry. No, we should also think about what bacteria. I think there's a lot of work that still needs to be done on the colonization potential of bacteria and especially also the link between the person and what can colonize. Because when for example, in many of these next generation probiotics, these are bacteria that are sourced from the gut. They have the potential to colonize the gut, and so let's keep the commercial part aside here. Purely from a scientific point of view, there is indeed a potential that if you give such bacteria, that they will colonize. But we don't, at this stage, we don't really know when they will colonize, with whom, in what context, because likely the reaction is going to be personal, and that some bugs will colonize in some individuals, but not in others. And it will also depend on the state of the microbiota at the moment of procurement. So there's a lot of factors that play into that idea of something colonizing, yes or no, exposure in the past, etc., etc. I don't think the word is out yet that it will not or will always happen. I think the answer is somewhere in the middle there.
Yeah. So another question. You alluded to statins having a protective effect, if I may condense that remark, putative protective effect. Obviously, there is the Groningen study. In the Groningen study, which is, of course, as in Flanders, also a cohort study, they showed that methylformamide being used as a medicine in old age diabetes also has a protective effect on general aging. Did you also see that, or did you just only see the statins?
This was a specific study that focused on the statins. This wasn't done this was done in a targeted cohort in a European project, focusing on the trajectory of let's say obesity to metabolic syndrome to cardiovascular disease. And so we specifically in that study only looked at statins, so I can't say anything else about that.
Okay. So that will follow later, probably. And then my last question to you, Jeroen, and then we can proceed to the next speaker. And any other questions to you can be asked after the wrap-up. The last question to you is, do the enterotypes, as you described them, need different diets?
Mm-hmm. Yeah, so that's still, I mean, I've been working on enterotypes now for 10 years, and still we don't have a 100% idea of what's the sources, the causes behind it. But we are convinced that diet is one of the driving factors. For example, there's very strong associations between the abundance of Prevotella, which is one of the driving organisms of one of those enterotypes, and fiber consumption. Also, I think we find associations between the carbohydrate consumption and the Bacteroides enterotype. So we do think diet is one of the more important drivers there, except for the B2, because there in the B2 we think it's more a dysbiosis, so that's more of a disturbed enterotype. But in the normal, let's call them enterotypes, diet is a player. But then again, am I at this moment able to tell you eat this and then I'll shift you from A to B? We can't at this moment. We still need to do many of the experiments before we really can do that. Those types of suggestions.
Great, and luckily you're still a very young scientist, although you're very established. So I look forward to the next 2 decades of work where you will elucidate this. A great talk, Jeroen. Thank you very much. A great talk as always, and also thank you that you will stay on board for the answering the questions in the wrap-up. So thank you very much. The next presentation is by Robert-Jan Brummer. Robert-Jan Brummer actually is Dutch, but he forgot about that because he lives in Sweden now for a long time. And he has been involved in establishing and growing the medical faculty in Örebro in Sweden. He's a very modest scientist, but I can tell you that was a Herculean effort on behalf of Robert-Jan. He's been working especially on the gut-brain axis in the context of the intestinal barrier. So that means that our intestines are a selective barrier. Not everything that you eat will get into your body. And that selective barrier has been the subject of the studies that Robert-Jan has been doing for the past 30 years, I think, not by now. And I welcome him. I really like his talks, and I'm sure you will enjoy what he has to say. Robert-Jan, the floor is yours.
Thank you, Erik, for your introduction. I will try to give a perspective to the gut-brain axis from my point of view as a gastroenterologist. But I have to say also that I've worked more than 6 years in psychiatry as well. So I'm also an amateur psychiatrist and have that perspective as well within the gastroenterology. As you said, I'm living and working now in Örebro the last 12 years. And just to make clear where Örebro is, because we have not that many criminals in the city, so it's not a very well-known city. It is about 200 kilometers from Stockholm, west of Stockholm, and is the newest medical faculty in the country. And actually we are in about the 400 position at Times Higher ranking, and about the 140 in citation index in the world. Only 20 years old. So I work as a researcher, but I'm working also as a clinician still, 1 day a week. So that will actually also color my perspective. So we start again with the gut-brain axis again and again. And I use very often the metaphor of music for my patients. So it's a bidirectional interaction between gut and brain, and you can see the brain as the pianist, the gut as the piano, and then actually your gut function is the music from the piano. And what I want to express with that it is not very often that there are a sort of anatomical failure, which is wrong, but it is a functional failure. So if the pianist is distracted or has just a bad sleep, he will perhaps not listen to his own music as well, and then the music as such is not that good. And that is what with the brain-gut axis as well. And it could also be that just for the piano is a little bit untuned, and then you get disturbed as a pianist by hearing that, and then it gets worse. So it's this interaction. And actually you can say that the scores of what you're playing, that is the food, what you're eating. So I think that is especially important to give this perspective that there's not anatomical problems very often, but more a functional perspective. So once again, what are the big players in the gut-brain axis? The content in the gut, which we have discussed already, the mucosa so the first barrier in the gut to the body, which is in number 2 here with all the cells there and very specialized cells as well. And then what already Erik alluded to is this very special enteric nervous system which we have and is connected to these special cells. And this enteric nervous system, especially by autonomic nervous system, is connected to the brain, number 5. So that is the more nervous route of communication. And then you have also the humoral route, the route by blood, which is directly taken up, passing the liver, metabolize it, and then if molecules are very small, they can pass the blood-brain barrier and pass from 4 to 5, and by that way enter the brain, and then the way back as well, which goes by hormones produced by the brain or other substances, as well as the vagal nervous again. So this is the interplay between the gut and the brain, and we will come back to that later on to go more in detail. So as a researcher, it's quite nice that it is so complicated, because here you see just a number of the many mediators of this bidirectional signaling. And of course, I will not go into detail, but I will just take one there, because we already spoke about serotonin. And yes, serotonin is important. We know that of all serotonin, about 3% is in the brain, 95% is in the gut. But, and that is very often misunderstood, the serotonin produced in the gut will not enter the brain. However, the tryptophan, which is the substrate of serotonin, that passes the blood-brain barrier. So the tryptophan in which you eat by proteins and is in your amino acid pool, that is important for making serotonin locally in the brain. And I will come back to that because that is important when we discuss also inflammation. Because the synthesis of serotonin out of tryptophan is affected by inflammation and stress. And that has clinical consequences. I will explain that later. So just to show you an experiment which we did for many years ago, but is still quite instructive and shows the importance of serotonin, is that we blew up a balloon gently in the colon in both healthy people as well as IBS patients, and then looked at what happened in the brain by, at that time, not that sophisticated for today, functional MR camera. And you see some things happening in the brain there. We did in exactly the same people, the same experiment, but we had artificially lowered their tryptophan level and by that their serotonin production. And we saw that there happened other things in the brain. And it's not only important where you have your activity in the brain, but it is even more important how the networking is going in the brain. So we could show that exactly the same balloon inflation in the gut by just having a low level of tryptophan and serotonin just changed the response in the brain, which is actually a direct proof of the importance of serotonin in this and the gut-brain axis. So that about serotonin. Jeroen already spoke a lot about the gut microbiota. And so what is the role for a gastroenterologist in the gut-brain axis from the gut microbiota? And we know that they have this own separate ecosystem, their own genome, and that will also, Ted Dynan will express that also. But I think we should not lose the perspective that these first years of your gut microbiome are pivotal for the rest of your life. So even if you have the same, if Erik and I would have the same microbiota today, but we had a different microbiota in our first 3 years, our immune system and our gut system, barrier system, has matured in a different way. So your gut microbiota of today is not saying everything about your function in the body. It's also how actually that changed during the first years of your life. And by that way, we have to be a little bit cautious and careful in interpreting what aberrant microbiota composition of today means for disorders. We have to have this life course perspective there. Longitudinal cohort studies can help with that. And this should also be taken into account when we talk about gut microbiota and brain function, of course. So how can gut microbiota affect the brain? And the different speakers will allude on that differently. But importantly, you can actually separate two actions. The direct action why a microbe directly acts to a receptor or whatever in the mucosa, and that triggers something. Or by what these microbiota produce, like, as Jeroen told us, these neuroactive substances. Ted Dynan will also speak about that. But also, of course, the butyrate and other, all sort of metabolites produced by the bacteria are important and affect the brain. And how can you do research on that? And therefore, I just show you an example of research project which we just have finished, where we got into detail how to look at the effect of butyrate. Already Jeroen stressed the effect of butyrate for our body, also for our brain. And here we did actually an experiment where we very systematically could study the effect of butyrate. So we introduced a tube by an endoscope in the lower gut, and then actually take biopsies before, so small specimens of the gut, which we can look at, also look at barrier function, and then immediately after we exposed a part of the gut to butyrate solution, and then we have actually closed this segment by 2 balloons so that the butyrate solution stays between those 2 balloons. And after 1.5 hours, we deflate the balloons and take the biopsies of the area which was exposed to the butyrate. And one of the things which you have not spoken about yet is that, for example, people differ in their way they can take up the butyrate. So it's not only important how much butyrate you produce, but also are you able to take up that? Are you able to utilize it? And we see, for example, that patients with irritable bowel syndrome have an inferior capacity to utilize the butyrate. So one of these things which we did here is looking at, if you give butyrate, can you upgrade that and can you actually restore that? I will not tell you the results yet because they are not totally proven. We have to look at that. But that actually, by this kind of research, you can look more in detail. And then also we mount these small specimens, which we take on the second picture here. You can see how we take these biopsies. We can mount this in a certain chamber and can look in detail on the barrier function of these, this material. So I already said barrier function. So where the barrier function is a very important part, and Erik already introduced me by my view on the barrier function. So what is the barrier function? As you can see here is that the barrier function of our gut is actually more or less the firewall of a computer to your internet. What the gut wants to take up important nutrients from what you're eating, but of course should avoid taking up very dangerous compounds. And that's exactly the same as a firewall in your computer. And you see that you can take up your compounds by different ways, partly through the mucosa, through the cell, which is the normal way, but also in between cells there is a little bit of space, and compounds can actually pass there. And this interspace should not be too big, because then all sorts of compounds which you don't want to pass can pass, like remnants of bacteria. And what they do is they trigger the system below there, they trigger separate cells there, specialized cells, and these cells can trigger the enteric nervous system, and by that way trigger your brain, because the enteric nervous system is connected to your brain. So this is how an impaired barrier function can have a direct effect on the brain. And there are quite a number of intestinal, or quite a number of disorders which are characterized by an aberration in intestinal barrier function, like, for example, inflammatory bowel disease, but certain people with irritable bowel syndrome as well, metabolic syndrome, overweight, aging, stress, strenuous exercise, just 1.5 hours on a strenuous exercise, you have quite a deteriorated barrier function. Dehydration, we recently showed, also only dehydration by such also give you an impaired barrier function, etc., etc. So this is an important thing to consider. And what it does with your gut and then with the brain is that it gives you this local immune response. This local immune response can become a systemic immune response. Mast cells, those cells which are involved in allergic reactions, are also activated and produce histamine and tryptases and so forth, and actually iterate this system by even making your tight junctions between the cells worse, and then actually decrease your make even the immune or the barrier function even worse. And that gives, at the end, this triggering of the vagal nerve to the brain. So now my most complicated slide, and that is try to integrate this with microbes, barrier function, inflammation, serotonin, stress, and mental health. How is that now interrelated, and what is inflammation there doing? And actually what happens is that if you have inflammation in your body or in your gut, as well as in your liver, what you will do is you will trigger processes where tryptophan is not longer metabolized towards serotonin, but is going to other compounds. And that actually, so what happens is that if you have an inflammatory activity in your body, you will produce less serotonin from the tryptophan available. And that is the reason why you can get depressed when you have an inflammation. Also backwards also, it works as so. If you have a lot of stress, you will be more prone to inflammation. I will not go into detail now on that. So, but this is the reason how serotonin is actually linked to the gut, the brain, the microbes, stress, and so forth. So if you look at irritable bowel syndrome, how do we as a gastroenterologist can work with that? Well, one of the things there is that some patients with irritable bowel syndrome have actually a deteriorated barrier function. So that would be the primary point to work on. For example, people with diarrhea, predominant irritable bowel syndrome. So for example, by prebiotics or probiotics inducing butyrate production, you can try to repair that barrier function, and by that way actually change their gut-brain axis and also make them feeling better. Inflammatory bowel disease, we have worked a lot with the serotonin there, and I've had a lot of IBD patients which I gave antidepressant drugs, not because they were depressant, but just also for treating their inflammation, with actually quite some success. Very often you see that they have also some signs of depression as well. If we look at microbial therapy, what are the different options? Well, we spoke about prebiotics and probiotics. Of course, antibiotics, and in certain cases we give antibiotics, special antibiotics, to improve the gut flora, which sounds very strange, but it can work, as well as fecal transplantation or fecal transfer. And we have quite some experience with that, but I always say be very, very careful with fecal transplantation. So how does it work with these pre and probiotics? One thing we have not discussed yet is actually the small bowel. We all think that it has everything with these microbes have with the large bowel to do. But the small bowel is overlooked. And my personal belief is that the effect of probiotics perhaps is as much a small bowel effect as well as a colon effect. The small bowel is not used to many bacteria, and if you give this high dose of probiotic bacteria, you will absolutely get an immune effect. And how much is the direct microbe-host interaction involved, or how much is it the products like the butyrate, and what kind of compounds are important? And how can we enforce this intestinal barrier in a way, not only perhaps with pro and probiotics and this anti-inflammatory effect? So there are a lot of ways how pre and probiotics can affect gut-brain interaction. We know a lot, but we need to know more about that. Also just to show you what we can use in practice is that we, for example, use now functional magnetic resonance imaging to look actually at the effect of probiotics in the brain. And we are actually can show that even a shorter course of probiotics, you can really see some changes in brain function just on the level of functional brain imaging. So what are the potential functional applications of probiotics? Of course, these effects on stress, and Ted Dinan will speak about that, on the effect of depression. You can also try to modulate satiety. We have not in this seminar so much about that. In the long term, perhaps also preventing and delaying the neurodegenerative disorders such as Parkinson, multiple sclerosis, and so forth. But we need to personalize it. We can't only look at the group level. And this is the great hinder so far, to look at the personalized, what is working for each individual best. And then, after all, we can speak about prebiotics, antibiotics, fecal transplantation, but don't forget the importance of the whole diet, because that is what all the bacteria have to eat every day, and that is what you eat. And by that, I would like to conclude my presentation for today, and I'm happy to answer on questions.
Professor Brummer, Robert Jan, great story as always. Happy to see Swedish breakfast here with all the berries neglected, like the nuts and the berries. We have a number of questions. First question is, what happens to all the serotonin that is produced in the gut but can't cross the blood-brain barrier? Is it good for anything, or is it just for nothing?
Mm-hmm. Yeah, serotonin is very important for gut motility, for example. So serotonin is locally in the gut very important, and also has its effect on the enteric nervous system. Yeah. So it's a direct relationship between serotonin and the enteric nervous system. So to keep the gut healthy, you also need a specific load of serotonin.
So a happy gut is a healthy gut, or a healthy gut is a happy gut? But
I wouldn't say that not always it's more serotonin is the better. We also know from research that in certain inflammatory circumstances you see a little bit more of serotonin. But it's the serotonin activity which is important, not only the concentrations of the serotonin.
On that note, is there such a thing like a mood food, like a food high in tryptophan?
The problem a little bit is that if you increase tryptophan in your diet, it can be just metabolized by the liver and that just not enter the brain. So it's not that easy. But it has been shown that, for example, if you at the same time increase your insulin level by actually, well, it's by changing your amino acid pool, actually, you can have an effect. So in people which are under stress, it seems that an increase in tryptophan can have an effect.
Okay.
But there is not that much research on that.
Yeah. Challenging you on your diet slide, which specific prebiotics could be useful in IBS?
What we think is that, and that is actually also a question of a good and a bad balance, because if you just increase your dietary fiber, you will probably increase also your butyrate production, which is good.
Yeah.
But by that fermentation, you also increase a lot of gas production, and that IBS patients don't like. So this is one of the dilemma we have. So what you want to have is a fiber which gives a lot of butyrate, for example, with a low amount of gas production. And one of the things is that also it should not be only on one stage in your gut, because you need butyrate over the entire colon.
Yeah.
And if you have just simple prebiotics, they will just be fermented in the upper part of your colon, and they don't reach the lower part. So it is more complicated than just a little bit of extra butyrate somewhere.
Yeah.
And actually a fecal sample doesn't measure that at all, because that is just what the body leaves.
Yeah.
And actually you could even say that if there is a lot of butyrate in your fecal sample, you have not taken up it.
Yeah.
So perhaps you are these IBS patients who have a problem with taking up butyrate, and then you have a higher level in your features.
You did not elaborate on this in your presentation, but I know that you also take poop samples in the gut, not from the poop that comes out, because as you say, that's largely an end-stage poop,
Yeah.
and it doesn't say anything on the upper colon, let alone on the lower intestines, on the small intestines. But you don't just take biopsies of the tissue, but also biopsies of the poop. I think that's a very important point, and it argues also for working in humans, I guess, because you
There you have also a problem that in that case, if you want to do it right, you have not to rinse the colon. What we always use when you do a colonoscopic investigation. So we do actually investigations where we do not prepare the subject and go in with the endoscope in a colon, in an intestine full of poop.
Yeah.
And that's the only way to do it if you want to do this kind of research.
Great. Could you give an example on where an antibiotic course would actually help the patient in re-establishing a flora that is beneficial, or is that difficult?
Yeah, well, the classic example is Clostridium difficile,
Of course.
where you have an ecosystem which actually favors the Clostridium strains which produce the toxin. And then by changing the ecosystem in some way, you can actually get a better balance between this Clostridia and the other bacteria and decreasing the load of toxin. But that's not always effective, but it is the first way of treatment. IBS patients we sometimes treat with a local antibiotic, rifaximin, trying to improve the gut flora, but it could also help in reducing small bacterial overgrowth. For example, if you have an overgrowth of a bacteria in the small bowel, you also want to treat that.
Yeah. Is it, I mean, you asked for this question. I love the question. You said you had some psychiatric experience. I know you do. Not personal experience, but with your patients, of course. But is it advisable to tell a depressed person to change his diet?
I would say that there is nothing wrong with changing it to a better diet. We know from studies that in Sweden, we think that patients with depression or subjects with depression, not everyone is a patient, they are about we would guess about a 25 percentile of a good diet. So they eat worse than the average Swede. So there is a reason to improve diet. And we think that you can, by a Mediterranean diet, or what we call a healthy Nordic diet, which is the same sort of principles like you see here on the slide, you are able to improve. However, there are these studies which also has been shown earlier by Jeroen also, and it will be shown by Ted Dynan also. But we are not the proof. There are so many other factors which play a role in this kind of studies. So we are now starting a study where the people get everything made beforehand, and then so there is no bias by that you choose the one diet or the other, but you have to eat exactly what you get.
So it's also a personalized diet.
Yeah.
So another question, completely from left field: is it possible that the effects observed in the lung COVID, generally called foggy brain, are somehow related to the gut microbiota?
There was, I think it was yesterday or the day before yesterday, there was an item on Medscape Europe on that, actually. Actually, COVID is not different from, in that case, from any other infection, viral infection or whatever. And we know that you can get this sort of long-term consequences. We know we have post-infectious IBS, and post-infectious IBS has to do with a change in barrier function and so forth. And what we see actually, if we look at the bacteria, the gut flora in post-infectious IBS, they are not that aberrant anymore. They're actually more normal than a standard IBS patient. But there has happened something before which has triggered their immune system, their mucosa, and they are still hyperactive. We see increase of certain lymphocytes, for example, in the mucosa of these patients. So yes, probably during COVID your barrier function is challenged, and how you individually respond to that challenge can make it different. So there could absolutely be an association, but so we have to look more in detail about that in long-term COVID. But I would say it's not different from many other infectious diseases.
Which we have a lot, because clinically we test for at least 20 pathogens if you have a lung respiratory infection. So. Great. Can the Nordic diet also be advised to Mediterranean people, because they will have a different microbiota, so their microbiota might have different needs, or
Yeah, there could be. Honestly, I don't know, because that's right, we haven't tested. There are tests of Mediterranean diet in other people, but not the other way around. I would say that I think the kind of food is quite similar.
Yes.
The kind of fibers are quite similar. The difference is that you get your more omega-3 fatty acids by fish, for example, perhaps than in the Mediterranean diet, and there you get a little bit of other non-saturated fatty acids. And I think the berries and so on are more pronounced in the Nordic diet, with their antioxidants and so on.
Yeah. And then my last question, could brain food also help people with Alzheimer? That's a good one before the break.
Yeah, but you can say so that I would give a general answer that in Alzheimer you have a compromised brain. And you could hypothesize that a compromised brain would perhaps react better on a healthy diet for your brain as well, than a non-compromised brain. So yeah, the function which is left there, it perhaps could help. And what we think is that if you by diet can decrease inflammatory action, inflammatory activity, both in your system as well as in your brain, that will have, of course, a positive effect on neurodegenerative diseases, because they are characterized by neuroinflammation. Yeah. Their degeneration is initiated and associated with neuroinflammation. So theoretically, yes, but we have to show.
Yeah, and we have to be careful, because there were products out there a couple of years ago that took all this.
And that is with nutrition always the big problem, that you see in epidemiological studies an association between something and a disease, and then you try to revert it, and then it doesn't work.
Exactly.
Like the vitamin A trials in lung cancer and so on and so forth. So we have to be very careful to translate epidemiological research and our hypothesis to treatment.
Yeah, but okay, last question from me then. In your experience, is there a relation between people that obviously don't look like metabolic syndrome and Alzheimer, or is there no relation in your clinical eye? Let's say the clinical eye, is there a diet component if you look at the patients, or did you never?
I think you have to look at an individual there, and then the things we see on group level are not always true for the individual. So we have to be very careful there. Yeah.
Okay.
Yeah.
Well, I hope you're ready for the next part of the symposium. And that is a very renowned speaker from Ireland, Professor Ted Dinant, who will tell us much more on the importance of the microbiota in stress-related disorders. And he will try to explain how our gut microbes communicate with the brain and what these microbes play for a role in mental disorders, and how this communication then works. So we're all very excited to have you here today. Thank you very much for that. And the floor is yours, Professor Dinant.
Thank you very much for that introduction. It's a pleasure to be here. So what I would like to do is illustrate how gut microbes can influence our mental health, and how, in fact, we might be able to target the gut microbiota in disorders such as depression or anxiety. If we could have the next slide, please. Within the intestine of the adult, we have between 1 kilo and 1.5 kilos of bacteria, and there are over 1,000 species of bacteria within that microbiota. So it's an enormous volume of bacteria, and that bacteria can influence the brain in a variety of ways. Now, what determines the microbiota that any one of us actually has? What determines the composition of that microbiota? Well, the first big determinant of our microbiota is how we were born. A baby born by caesarean section will have a far different initial microbiota to a baby born through the mum's birth canal. The baby passing through the mum's birth canal will pick up lactobacilli as it passes through the birth canal, whereas the baby born by caesarean section will develop its initial microbiota from bacteria on the skin of the mum, the skin of the doctor, and from the general environment. Now, over the first 2 to 3 years of life, those two microbiotas do tend to converge. So by years 2 to 3, the baby born by caesarean section and the baby born per vaginum will have a very similar gut microbiota. But we do know that caesarean section rates are on the rise dramatically internationally, and we know that babies born by caesarean section have higher rates of asthma and allergies, and this is probably related to the initial gut microbiota. Now, other factors that determine our microbiota would be our diet is obviously a dominant regulator of our gut microbiota composition. Exercise is very important. Professor Brummer has mentioned antibiotics, and obviously they can dramatically impact the gut microbiota, but it's estimated that about 80% of all the drugs that we currently prescribe in medical practice impact the gut microbiota in varying ways. So there are a variety of factors that determine the composition or the architecture of our gut microbiota. Now, stress is a phenomenon we all experience, and clearly up to a certain level, stress is a good thing. We wouldn't perform well if we weren't exposed to some degree of stress. But beyond a certain pivot point, undoubtedly stress results in a variety of psychiatric and medical disorders. And Hans Selye, who's on the right-hand side of the slide there, was one of the first people to show that cortisol, the main stress hormone, had a very dramatic impact in a variety of medical conditions. And of course, it was elevated as a result of stress. And during the COVID epidemic, we see far greater numbers of people reporting stress as a problem for them. Under normal circumstances, if you survey the general community, about 10% of people report problems with stress. But during COVID, most surveys suggest that up to 40% of people have experienced stress-related symptoms. So the main psychiatric disorders we see which are related to stress would be depression and anxiety related conditions. Now, when we talk about gut microbes influencing the brain, they communicate with the brain in a variety of ways. The vagus nerve is obviously one important route of communication, but as Professor Brummer has already pointed out, there are a variety of chemical substances that are produced by bacteria or are released from the gut that circulate in the blood and can influence brain function. Short chain fatty acids are synthesized by bacteria, short chain fatty acids like butyrate, and they will influence many organs, including the brain. We've also heard how tryptophan, the precursor of 5-HT, is something that is released from the intestine. Now, traditionally, the view has been that it all comes from the diet, but of course, we know that tryptophan can be synthesized by bifidobacteria, so that's another source of tryptophan entering the bloodstream. Now, I find as a psychiatrist, I think it very interesting that most of the common neurotransmitters can be produced by bacteria. So when you look at serotonin, noradrenaline, GABA, acetylcholine, they're all produced by microbes in the gut. I'm not suggesting that they go to the brain and influence the brain directly, but I do think it's intriguing that microbes are capable of producing so many of these neurotransmitters. Now, I regard the large intestine as a bit like a big fecal fermenter. It's a big fermenting organ that produces a variety of molecules, and these molecules enter the bloodstream, and some of them have a significant influence on brain function. Now, 4 or 5 years ago, we published this paper. I was interested in seeing what sort of microbiota do the patients who attend my clinic with depressive illness have. So we took a group of depressed patients and we took a group of age and sex matched healthy controls. And if you look at the right-hand side of the figure, you see that the microbiota of the depressives is very different from that of the healthy controls, and that overall the depressives have a less diverse gut microbiota than do healthy subjects. Now, we all know you cannot change the genes in your cells, but we can certainly change the genes in our gut microbiota, and we can do this in a variety of ways. So, what are the ways in which we can change the genes in our gut microbiota? Well, there are easy ways and there are very radical ways. Fecal microbiota transplantation is obviously a very radical way. The only clinical indication for that right now is for treating C. difficile infection. But clearly, one could use probiotics, one could use prebiotic fibers, one could have a radical change in one's diet, and that will clearly alter the gut microbiota. And as I've already said, undoubtedly, many medications influence the gut microbiota. Many patients I see at my clinic are on proton pump inhibitors for treating acidity or peptic ulcers, and they have a dramatic impact on the gut microbiota. Now, a few years ago, we introduced a concept into the literature that seems to have caught on, and it's the concept of a psychobiotic. And we defined a psychobiotic as a bacteria which, when ingested in adequate amounts, has a positive mental health benefit. So I want to stress the point that not all probiotic bacteria have a positive mental health benefit. In fact, many of them do absolutely nothing from a mental health perspective. But this is one study we did with a biflongum, and it was a study in which we looked at stress responses. It was placebo controlled. We looked at stress responses, both psychological response and also cortisol release, cortisol being the main stress hormone in humans. And what we found was that in contrast to placebo, when subjects were taking the biflongum, they reported themselves as feeling less stressed, and there was a concomitant reduction in cortisol levels, the cortisol we measured in the saliva. So this biflongum seemed to reduce stress levels and reduced the cortisol, which is the main stress hormone. Now, this was a study we conducted in students where the students were examined over 2 semesters, and it was during the examination period. And in one semester, they received a placebo, and in the other semester, they received a biflongum. So everyone was tested over 2 semesters, and we were looking at sleep quality. And what we found was when the subjects were taking the biflongum, the sleep duration was increased relative to that seen when they were taking placebo. So this now was during a stress period. There are at least 2 other studies in the literature recently also supporting the fact that certain probiotic strains do seem to improve sleep quality. Now, this is another study of exam stress from the Takeda study, which was published 4 or 5 years ago. And it is again a placebo-controlled study using Lactobacillus casei, the Shirota strain. And what they essentially observed was the subjects were examined, or sorry, the study was conducted during an exam period. And the question was, did the placebo have an impact similar to the probiotic, or was the probiotic more effective? And if you look at the right-hand side there, you see that in fact that the probiotic, this Lactobacillus casei, attenuates the cortisol release. The subjects felt less stressed and they reported less physical symptoms when taking the probiotic strain. The next study was a study in depressed subjects. Whoops, sorry, could we go back to the previous slide, please? Yeah, no, the next one, please. Yeah. Now, this is a study of 18 depressed subjects, and what you see here are the Hamilton rating scales and the Beck depression rating scales over a 12-week period. And what you see is when the subjects were treated with Lactobacillus casei, you see a reduction in the Hamilton score and a reduction in the Beck score. Now, they're 2 of the standard score ways of rating depressive symptoms. And as you can see, the probiotic strain seems to be capable of reducing depressive symptomatology. Now, I only put this slide up to demonstrate that not all probiotics actually do anything from a mental health perspective. This was a probiotic that we had very high hopes for, but when we tested it, it had no impact whatsoever on any mental health, immune, or endocrine symptoms. This is a study of a Lactobacillus rhamnosus in 423 women. Now, the women were randomly allocated to receive either the Lactobacillus rhamnosus or the placebo from the second trimester of pregnancy until 12 weeks postpartum. And what one essentially found they found in this particular study is that the women who received the Lactobacillus rhamnosus from the second trimester of pregnancy postpartum, they had far lower depressive and anxiety symptoms. So it does seem as though this particular probiotic impacts on depressive symptoms during and in the postpartum period. Now, this is a relatively recently meta-analysis of studies of probiotics in patients with depressive illness, and it concludes that overall that probiotics are effective in treating depression and that multiple strains seem to be more effective than single strains. Now, I would add that we do need larger scales placebo-controlled trials. This is based on a meta-analysis, but it's an interesting conclusion. Now, prebiotic fibers are found in a variety of vegetables. Jerusalem artichoke is a particularly high level of prebiotic fiber, and prebiotic fibers promote the growth of good bacteria. So if one wants a lot of good bacteria, one takes in prebiotic fibers, undoubtedly one will promote the growth of good bacteria. This is a consensus document, an EU consensus document funded by FP7, and it's essentially giving guidelines for the dietary management of depression. Now, it's not suggesting that we manage depression with diet alone. Medication and psychological therapies have a major role to play. But undoubtedly, patients who take high levels of fruit and vegetables in, who have high levels of fermented food and a high intake of fish, are more likely to respond to psychological and pharmacological therapy. So I think diet has a role to play in the management of depression. And this study demonstrates it, I think, very convincingly. Here is a study from Felice Jacka at Deakin University, where she took a cohort of depressed subjects. Half of them were randomly allocated to receive social support, and half of them were randomly allocated to go on a modified Mediterranean diet. And this is the changes in the Hamilton score, the depression scores over a 12-week period. You can see that those who went on the modified Mediterranean diet had a very dramatic drop in depression scores, far higher than that seen with social support. So it would seem as though diet does have a role to play in managing depression. Now, this is a case vignette. Case vignettes, when you draw conclusions from them, they must always have a health warning. And I put a health warning over this because vignettes are no substitute for doing large-scale placebo-controlled trials. But this is an interesting patient because this lady presented to me she was a 44-year-old married lady with 2 teenage children. And she was sent to my clinic because she had been depressed for about 12 months, so quite a long period of depression. She had had one previous episode of depression following the birth of her first child. Now, the current episode of depression did not seem to be precipitated by any psychosocial factors. She seemed to have a reasonably happy marriage. There was no major financial problems in the family or anything else. And there was no family history of depression. Both her parents were alive and well, and she has 2 sibs, neither of whom were suffering from mood disorder. She had underwent a course of cognitive behavior therapy with a very good psychologist, and it had not impacted on her symptoms one bit. At the same time, she had been prescribed very adequate doses of escitalopram and venlafaxine, 2 very effective antidepressants. Neither of them had any impact. Now, when I saw her, I made a number of changes. First of all, I suggested that she begin an aerobic exercise program. I suggested that she make certain dietary alterations, and she also commenced on a lactobacillus probiotic. Now, what was interesting here is that there was a major improvement in her symptoms over a 4-week period. This is a woman who had been ill for 12 months, and now over a 4-week period, with exercise, with change in diet, and this lactobacillus probiotic, she undoubtedly improved. And even I followed her up at a clinic, and even 6 months after these alterations, she remained free of symptoms. So it's an interesting observation. I'm not suggesting one makes major conclusions from it. At the end of the day, I believe that antidepressants and CBT have a very significant role to play in managing depression. But I do believe that diet and probiotic supplements can have an important role to play in some patients who are depressed. So, in conclusion, ladies and gentlemen, I think that gut microbes are essential for normal brain function. The gut microbiota is dysregulated in depression. There's also evidence it's dysregulated in some anxiety disorders. I think that psychobiotic bacteria, they're bacteria that have a positive mental health benefit, acting through the stress system can impact the symptoms of depression and anxiety. And I do think that diet is very important when we're treating depression. And I always give my patients, whatever about a prescription for an antidepressant, I give them dietary advice and advice in relation to exercise. Thank you very much.
Professor Dinan, thank you very much for a very clear and concise, way within time presentation. Thank you also for that. We do have immediate questions here. One is, of course, what about a placebo group? If you do a case study, there is no placebo. Are you also willing to treat the next patients in this way, or will you start a larger clinical trial on this?
Well, I think that, really to demonstrate efficacy with any therapy, whether it's a placebo, whether it's a probiotic therapy or whether it's a dietary intervention, I think one must have a placebo group. So so really most of the large or the studies we design, we try to incorporate a placebo group, and I think that one always draws firm conclusions only when one has a properly designed placebo-controlled trial.
Yeah. The next question from the audience is, could you revisit the point where probiotics trigger the brain and especially elucidate something on the mechanism? Is that through the nervous vagus or hormones or whatever?
Certainly. Well, certainly, one of the ways in which bacteria in the intestine influence the brain is through the vagus nerve. Now, we've published papers showing that certain bacteria can only influence the brain when the vagus nerve is intact. If the vagus nerve was severed for any reason, certain microbes cannot communicate with the brain. Other routes of communication are bacteria produce short-chain fatty acids like butyrate. They enter the bloodstream and can influence the brain via the bloodstream. Other important communication methods would be the production of tryptophan, which again enters the bloodstream and travels to the brain. The human brain has very limited storage capacity for tryptophan. So we need a constant supply entering the brain, crossing the blood-brain barrier in order to keep serotonin levels normal. There are other communication routes. Cytokines produced at the level of the gut can again enter the bloodstream and travel to the brain and signal across the blood-brain barrier. So there are a number of parallel routes. Some of them are nervous routes like the vagus nerve, and some of them are humoral or biochemical routes.
Yeah, the next question is, you elucidated the role of diet, but how large would this role be for mental well-being?
Well, I am firmly convinced that for people who suffer from depression, that treating them is much easier if they're on a proper diet. Many patients who come to my clinic with depression are on a diet of fast food or highly processed foods. Those foods are pro-inflammatory. It is much more difficult to treat a depressive episode with that type of a diet. So I would strongly advocate all of my patients, apart from taking the antidepressant that I might prescribe them, to change their diet and to have a diet that is, we've spoken of a Mediterranean diet. It doesn't have to be a strict Mediterranean diet, but certainly they should reduce their intake of highly processed foods because it really impedes recovery from depression.
In line with that question, and thank you for that excellent answer, one of the audience people is wondering how much probiotic supplements you would then have to take daily if you go with a better diet and a probiotic supplement.
That is a very, very good question. And one of the problems with a lot of probiotics, in fact, with most probiotics on the market, is we say that certain probiotics are effective in helping anxiety or helping depression. There are no dose response curves, or virtually none in the literature. Now, nobody would put an antidepressant on the market without doing a dose response study. But the doses of probiotics for many probiotics have been plucked out of thin air. So we really don't know what the optimal doses are at all. I mean, you have some probiotics at 10 to the 8 CFU, some at 10 to the 10 CFU. If somebody is on a lot of fermented food and in varied fermented food, are they benefiting from adding in a probiotic? One can't definitively say. But I think that, the more recent studies would suggest that the greater the variety of fermented foods or probiotics taken in, the overall better the better it is for one's general inflammatory status and probably mental health status as well.
So you see patients, and if you see patients and you say you should take more fermented foods, I'm sure that patients will look at you like, what are you talking about? I mean, do you give examples of fermented foods, or what do you do?
Yeah, I do. And like the reality is that if you go into any supermarket nowadays, I mean, you get a wide variety of yogurts. You have kefir, you have kombucha, you have kimchi. There are a whole variety of fermented foods you go into Lidl, Aldi, any of the supermarkets, they all have a good variety of fermented foods. And so I think, I find that more affluent patients attending my clinic are probably taking fermented foods, but unfortunately, most of my patients are not from an affluent background and do not take fermented foods. But yogurt, Greek yogurt, is very cheap to buy, and I would certainly recommend that people take as great a variety of fermented foods as is possible.
So the next question is, how does the gut microbiota diversity then relate to mental disorders in your view?
An interesting question. I think that, when you lose diversity, you lose the capacity to or you reduce the capacity to produce short chain fatty acids. If you lose bifidobacteria, you will certainly decrease the production of tryptophan, and we've shown that certain bifidobacteria, if ingested, can significantly increase the blood levels of tryptophan. So really, decreased diversity undoubtedly reduces the capacity to produce many molecules. And I would add, and something that, we all want to age healthily, and there is now overwhelming evidence in the literature to indicate that a loss of diversity in the elderly in particular is followed by frailty very rapidly. So really, aging healthily is associated with maintaining diversity, and loss of diversity is not just mental health implications, it is implications for the general physical well-being as we age.
Yeah, and the next question relates to that too, and it's what's the status of fecal transplants on treating patients with mental disorders?
At an exceedingly early stage, all you have in the literature are a few case reports. There's hardly a week goes by I don't get some email from an American wondering if they come to Ireland, can they have an FMT for their depression or whatever. But there is no indication at the moment for using FMT in people with mental health problems. There isn't any adequate data out there.
Yeah, we might add that due to safety aspects,
Absolutely.
fecal transplants outside of direct siblings and spouses and partners might also have a safety aspect there. So the do-it-yourself, do-it-yourself
That is
FMTs.
for sure.
Yeah. I think the next question is, I like it, but it is probably a difficult question. What kind of probiotics do you use yourself? But feel free not to answer this.
Well, some may have seen there was a paper by Sonnenberg about a month ago published in Cell, and it looked at it basically took a variety, quite a large cohort, I think about 64 subjects, randomly assigned half of them to go on fermented food and half of them to go on prebiotics. And what they found was that prebiotics did very little when it came to inflammatory markers, but that fermented foods radically reduced um, uh, uh, inflammatory markers, and that the greater the variety of fermented foods, the better. Now, I must say, throughout most of my life, I've eaten yogurt, but in recent times, I've added kefir, uh, occasionally kombucha, and one or two other fermented foods as well. So I'm a big believer that diversity is important when it comes to fermented foods as well. So personally, I do try now, I didn't before, to have a greater variety of fermented foods.
Yeah, and in my introduction, I put in a slide. We can make also these slides available, indeed, of foods that both are available, as you say, in the supermarkets. And the other one is, of course, that the range, as you said 10A to 1010 in normal language, that's 100 million to 10 billion microbes per dose. That range, as you mentioned, is also very important, and it's also therefore, I think, important that you also give a dose recommendation to people buying these products. If you can, buy the product with the highest dose. Of course, if you can't, you just buy the product because that's your variety. But within the variety, there is also, I think, a dosing aspect. And luckily, this is all published, so you can look this up. But it's not known to the general public, unfortunately. Thank you very much. For you, at this point, these were all the questions. And again, thank you for the excellent answers. I hope you'll stick around for a few more minutes, because we never know what other questions will come. We do have another question for Professor Raas in the Flanders cohort. And the question is, have you done any studies on kefir, which was now mentioned a couple of times, kefir use in the Flanders cohort? Jeroen, is he still there? Okay, well, we'll answer that question on the email. Sorry for the rest of the audience. And for Robert Jan is still here. In an interview, you said that you should not eat too much proteins because they can cause a disbalance in the gut. How much proteins should we eat each day, Professor Brammer?
Yeah, actually, the question already makes the problem clear, because it's not only the amount of protein you eat, it's also the amount of protein you absorb.
Yeah.
And the problem is that if you eat protein, you will always malabsorb a little bit of the protein. If you eat plant-based proteins, you will malabsorb much more of that protein. And that protein leaks to the colon and will be fermented by the colonic bacteria. And the problem is that most of these compounds produced by the colonic bacteria from these proteins are not beneficial for your body. Some of them are, some of them are not. So we make a point that just eating a lot of protein is not good for your colon, and by that for the rest of your body. How much do you need? It's hard to say, depends on what kind of, how much exercise, all that sort of things. But normally people don't need much more than 1 gram per kilogram body weight. And that is per kilogram body weight. You should, of course, look at how muscular you are and so on. But more, even 0.8 is very often enough for people. And if you, of course, are very obese, then you should reduce that, because your fat doesn't need proteins so much. So then it's actually your lean body mass which counts.
Yeah. Now, I know you're an athlete and you look, well, let's
quite
say...
skinny.
Quite skinny, thin, I was going to say. So are you on a caloric restriction diet, or are you restricting on everything, both?
No, I eat as much as I can, more or less, but I actually restrict myself on proteins.
Okay.
And the one other reason is I have only one functioning kidney also. So therefore I have to be, but I really try to look at not to eat too much protein.
Yeah. Okay, well, a clear answer to me. And then there is another question for Professor Dijnen. Professor Dijnen, do you think there will be more commercial probiotic products specifically aimed at mental health, like stress, depression?
I think that there are many different probiotics currently under investigation, and I do think that we will see within the next few years probiotics with significant data behind them. The problem at the moment is that there are companies out there touting probiotics as having mental health benefits, and there's absolutely no testing behind those probiotics. And they make outlandish claims, so it's very difficult for the consumer who goes into a health food store and sees a probiotic with some ridiculous claim being made for it. Maybe the claim is being made online or whatever, and there may be little or no data. So it's hard to kind of separate the genuine products from products that really have absolutely no data.
Yeah, agree. So on the other hand, as Robert Jan and myself, you are also involved in at least talking with industry on this, at least. So wouldn't you agree that there is this, surge in interest by industry, even the pharmaceutical industry, to revisit the effects of diet, also in terms of therapy, so that it might not be a food, but it might be an RX that is actually registered as a medicine?
Indeed, absolutely. I think there is certainly more interest, but and one would hope, I mean, as a psychiatrist, I would really like to see, an RX product reach the market, with really solid data behind it. And there are obviously, some very legitimate science-oriented companies out there, but they're competing with companies that have no scruples whatsoever and will make all sorts of outlandish claims. That would never happen with a drug. I mean, nobody's going to put a drug on the market and claim that it does X, Y, and a Z. They obviously would end up probably in prison, which would be appropriate. But unfortunately, when it comes to probiotics, you have very legitimate companies out there, and you have what I would consider cowboy operators, competing with them.
Yeah. Robert Jan wants to say something. Robert Jan.
Yeah, I want to follow up on that, because there is a problem with this kind of diet or probiotics or whatever if you compare it to drugs, because actually most of the drugs we give to our patients, only 40 to 60% of our patients react positively on these drugs. 40% is non-responders, and that is for antihypertensive or antidepressants or whatever. However, in the statistics, when you do the statistics, because of this strong effect in the responders, you get still a significant effect in your population. However, if you do something with an effect which perhaps only 20% of the people are responders, and it is not that strong effect either. It's very hard to see it and to see the signal from the noise. Yeah. However, we know, and Ted and I know all the adverse effects when we started with antidepressants. Most of these foods don't have these adverse effects. So there is another balance between the benefit and the risk, and the benefit and the effect. And our general statistics and our general way of analyzing this kind of things are not adapted to that. Yeah. We use still the pharma paradigm, and I think we should work on other ways of assessing the value and the benefit of these kind of products.
Yeah, so that,
and the same is also if you look on a we have counted in Sweden that if you decrease the blood pressure, if you decrease salt intake by 1.5 grams a day in every sweet, you reduce mortality by about 1,500 people a year.
Whoa.
I mean, no drug would be registered with this very small effect which 1.5 gram salt reduction has. But if something has an effect on the entire population, you have still an enormous public health effect. Yeah. If we can change food, and we have, although not the effect on mental health comparable with an antidepressant, but still an effect, and we can have that on the whole population, of course the effect on public health is huge. And we should have that into account.
Talk about huge effects. Yes. Another question for you, Robert Jan. We talked about diet, but there's another question now. That is, what is the role of exercise in mental health? And Ted Dinan also mentioned that. So you might both want to answer this. But can you say something on the role of exercise?
I think Ted is better equipped to answer that. We are not exactly sure what the effect is, but it has an anti-inflammatory effect. It has an effect on insulin, your metabolism, which actually can increase your tryptophan. I should say it another. Tryptophan is competing with other amino acids on the blood-brain barrier. Yeah. If you can use these competing amino acids in building muscles, yeah, you have less competition on the blood-brain barrier level there. So more tryptophan can actually come into the brain. That's one of the effects. So there are multiple effects. But Ted, I think you are better equipped to answer this question.
No, I think you've put your finger on the most relevant, biological effects of aerobic exercise. But I am a firm believer that aerobic exercise has powerful antidepressant and anti-anxiety effects. It's difficult often to encourage patients who are depressed to engage in vigorous aerobic exercise, but when they do, the majority of them benefit enormously. I think if we really were able to put into a capsule whatever aerobic exercise does, it would be a multi-billion-dollar drug, that's for sure. Aerobic exercise is powerful and potently antidepressant.
Great, Robert-Jan, yes.
And actually, if you put it in today's perspective, it would be interesting to see that if you perform aerobic exercise outdoors, you're not able to look at a screen at the same time. Yeah. Or you can, but you make traffic accidents.
Yeah, that's true.
And I think one of the things we see nowadays is that you have not this sort of micro-relaxation periods. People are not relaxing anymore. They are just when they have a moment to relax, they look on the screen on their iPhone. And one of the things with aerobic exercises, especially if you do it outdoors, you are just a little bit outside your screen environment, and that also has effects on your brain and your mental health.
We have a mixed public, a mixed audience today. Micro-relaxation, is that a microsecond? Is that a second, 10 seconds, a minute? What's a micro-relaxation for you? You have to give us some clue here.
Yeah, for me, it can be about a minute.
A minute.
For me, myself, but that's just for myself.
Yeah. But so the micro is not microseconds. No. It's micro hours then. Yeah. Okay, great. Two more questions. One is, there's a question on a list of fermented foods because of lactose intolerance. I understand that because, if you look at fermented foods in the supermarket, you usually end up with dairy products. We will provide that list. There are two excellent English language publications giving these lists. So we will provide this on Friday, when we also provide the link for today's seminar. I will make sure of that. And the other question, again, to Professor Dinan, and with that we will stop the questions, is a difficult question, I understand, but has the message of importance of diet to support treatment of psychological disorders been incorporated in the curriculum of medical students now, or are we here at a disadvantage?
Unfortunately not. I mean, medical students, I don't think that the medical curriculum has changed so radically since I was a medical student or Robert was a medical student. And the reality is that there is very little nutritional information provided to medical students. I mean, there's a little, but it really is not adequate at all. And certainly if you go to your average psychiatric clinic, most psychiatrists will never mention diet and they will never mention exercise.
I unfortunately I agree, and I know there are studies that say that usually the curriculum, the doctors are 7 years behind, and the curriculum is 14 years behind. So in that light, we still have a lot of work to do. On the other hand, you're both here today and volunteering to share this with a large audience. So in that light, I think thank you again, because obviously this is also an important step of informing the media of where we are going with this. A short wrap-up from my side, and then I have some household issues which I would like to mention. We started out with the fact that the gut microbe brain axis is very complex. I'm sure that for a lot of issues, that complexity has only led to confusion. So we were not here to convince you or to confuse you. We tried to show you where the field is going and which types of findings now help in understanding this. Is this leading into concrete products? Well, we feel yes. We see industry on all sides, not just the food industry, moving on this. And we also see that a lot of studies are now being done with commercial products that you can just buy off the shelf. The next slide, I don't know whether somebody can put the next slide. Yeah. No, this is not working. Okay, no. This. Well, the point of the next slide was very simple. There are a number of products off the shelf that have been tested in clinical trials. So with a placebo, in a randomized studies where patients are where patients are allocated in a random way and scored double blind. These studies have been taken together. In this case, in a forest plot. I will not explain the forest plot completely, but in the middle of a forest plot, you always see the line of no effect. That's the line in the middle. And then on the left, you have less disease. Left is less disease, so that favors the treatment. And on the right, you have more disease of the forest plot, so that favors the placebo. And these meta-analysis and forest plots give you a direct insight into the fact that as a whole, whether commercial products, yes or no, help in depression and anxiety, because as a whole, in your first impression, the blue dots should then be left of the line. And as you can see in this study, that is largely the case. Not always, because if it was always the case, we would have had other presentations today, which could be much more firm in their conclusions. But still, I think this is a very promising way of looking at all these clinical trials. And as Professor Dynan already shown, some interesting decreases in stress with students go hand in hand, by the way, in the increase in cognitive aspects. So these also are commercial products, readily available, and tested also for cognitive aspects. And so it's interesting to see that the same product can give both a stress reduction and a reduction, an increase of learning behavior. And physical symptoms. So also physical symptoms like abdominal pains. Now, we all know if we have a big exam or a big wedding that we can get abdominal issues. Some people even get diarrhea just before an important lecture. These things happen, and these abdominal discomforts also can be quote-unquote repaired or even prevented by either the correct diet or probiotic supplements. So for today, I would say that knowing where we are going is very important, and spreading the word is very important. We will continue in this line, and I'm very happy as a scientist and as a participant that Yakult is actually sponsoring these sessions where we can freely talk on, what is interesting us and what is keeping us busy, so to speak. Now, 4 things. One is there will be a link this Friday, September 17, of this seminar available for all of you. And at the end of the month, there will be a written report in English on the seminar. So That's worthwhile to wait for, I would say, to see what the written report says. That would really help you, I think. But if you don't have that much time, you can use the link which will be made available on Friday. Again, as I said in the beginning, all slides will be sent to you in a PDF. Importantly, not all figures are available because some figures might be copyright protected. Now, usually that's not a problem, but you do have to ask permission to the author for publication. If that permission is impossible, the next question would then be, do you have an alternative slide? Because most scientists have alternative slides that are not on the copyright and could then be used if you need them in your paper. So please be aware of that. And with that, and with no more questions from the audience at this point, I would like to close. If you have any further questions, please email them to the organizers and they will be answered. If not, I would like to thank all the speakers for their immense, clear and expanded knowledge and the translation of that into normal English. I really appreciated that. I would like to thank all the people from the technical support today. That makes our life a lot easier. So thank you very much for the technical support. And I hope to see you again next year in yet another symposium on the gut in relation to the next best, maybe in relation to the respiratory system. Maybe not. We'll see it next year. Thank you very much for your attention and see you next time.